Hi everyone, my name is Remi Geohegan and I work with baker's yeast as a model organism in the Kelley lab. Now you know the work of baker's yeast from the crunch of freshly risen bread or a cold beer on a hot summer day, but did you know they also mate for life? In a complex microenvironment, yeast lacking independent movement find their mates through the detection of chemical messengers called pheromones, released by mating partners. When a yeast cell senses these messengers in the environment, they will elongate, forming a mating projection called a Shmoo, named after the cartoon character you see at the bottom left side of my slide. If successful, the partners will fuse and form a new cell. This change in shape in response to an external stimulus is called polarized growth, and many different cells, including macrophages of your immune system, neurons in your brain, and cancer cells, exhibit polarized growth. Yeast are therefore excellent models to study polarized growth, as we can reliably induce this response in lab with purified pheromones. When yeast shmoo, the cell must orchestrate multiple programs to reorganize internal structures to build scaffolds for their projection, directing building materials to the site of growth, and prepare to fuse, all while tracking the pheromone gradient of its mating partner. Each of these systems have been well characterized in isolation. However, the crosstalk between systems remains poorly characterized. So how do we map the interactions of 110 polarity proteins? To map the polarized proteome, we imaged yeast cells with fluorescent tagged proteins known to contribute to polarized growth in response to pheromone. Now, yeast do not like to line up perfectly for picture day and sometimes overlap. To do this, we wrote two computer scripts, Shmoo North and Shmoo Morph. I will direct your attention to panel number 2 on my slide. Shmoo North detects shmooing cells that have a mating projection by recording measurements of the projection length, width, and location of the cell body. Shmoo Morph then takes this data and orients the yeast along the polarity axis, and this step is crucial as it facilitates downstream image analysis, starting with our ability to create a composite image of over 100 cells with the same tagged protein. We then take the fluorescent intensity data, which tells us where protein localizes in the cell, and feed the data into an average cell shape, producing the clean profiles you see at the bottom right of my slide. Using these profiles, we can determine which proteins interact by mapping how they occupy the same region of the cell. Not only does our novel Shmoo pipeline recapitulate known biological interactions, but we can also use it to help us predict novel signaling pathways and protein functions. The main takeaway of my talk is that when we figure out how yeast find love, we're really decoding how life communicates. So next time you have a beer, thank the humble yeast.